Welcome to my short video on autosomal barring, a lovely pattern associated with breeds like Fayumi, Campine and Sicilian buttercup chickens. It also occurs in the Westfälische Totliger, which is sometimes called the death layer chicken. Autosomal barring adds a black bar on individual feathers onto a pheomelanin or silver background. It's been suggested to be caused by the combined effect of dark brown extension and pattern genes, but I've also seen speculation that it's caused by a dominant autosomal gene specific for autosomal barring. I mentioned in a previous video that I wouldn't be surprised if there turned out not to be a specific gene for autosomal barring, and this indeed seems to be the case. Extensive work by Dr. Schwochow and colleagues reported in 2021 indicates that this pattern appears to be associated with the two Burchin alleles and possibly also the brown allele at the hypervariable melanocortin receptor gene on chromosome 11. These alleles are at the extension locus, which we have seen in previous videos. It is thought that banding may involve these alleles working in conjunction with the agouti signaling protein gene. This gene is well known in many animals and has already been shown to be involved in countershading in hens and chicks, which evolved to camouflage them in the wild. It also regulates estrogen production to produce sexual dichromatism, which is the difference in plumage between hens and cockerels. Together, the extension and agouti genes are involved in regulating melanin type switching, which I suppose is how barring may work. The same scientists have also showed that dark brown is not required for autosomal barring, but that it makes it more pronounced and easier to phenotype. Dark brown is thought to contribute to the regularity and clearness of autosomal barring, although it's not required for barring to form. It is likely that breeders have, perhaps initially unknowingly, selected for dark brown to create a more appealing phenotype. This has later led to the assumption that dark brown is required for autosomal barring, even though this isn't strictly true. Most surprisingly of all is that the pattern gene seems not to be associated with autosomal barring, with no evidence for it in the many birds of the combined breeding and molecular studies. The researchers even suggest, and I quote, that our study provides strong evidence against the previous assumption that a patterning locus on chromosome 1 has a predominant role for the presence/absence of within-feather patterns in chickens. Given their important assertion, we need to keep an eye out for further research and possible revelations about the genetics of patterning. There could be some changes in the patterning story on the way.